Paul mentioned, my name is Mary Scott. I am a faculty member in the Material Science and Engineering Department at UC Berkeley, and I also have a faculty staff appointment at the National Center for Electron Microscopy, which is a part of the Molecular Foundry facility at Lawrence Berkeley National Lab. So perhaps unsurprisingly, based on that background, you might deduce that my research group emphasizes using electron microscopy to characterize nanomaterials. And today I'm going to be talking about some work that we've done using both supervised and unsupervised approaches to use machine learning for electron microscopy data analysis. So a little bit more background, and by the way, these are probably the most zoomed out images that I'm going to show today. So in my group, we use electron microscopy to study nanomaterials, as I mentioned. We are particularly interested in nanomaterial evolution during synthesis and use. And to do this, we take the amazing facility that we have at the National Center for Electron Microscopy. So you can see this beautiful building here hanging off the side of the hills in Berkeley that has this panoramic view of the bay. That's the Molecular Foundry. And the National Center for Electron Microscopy is actually the one that's kind of behind these trees. So we have, regardless of our view being a little bit blocked and us being a little jealous of the facility at the Foundry, we have an amazing electron microscopy characterization suite at NCEM. And we also have our group at Hearst Memorial Mining Building on campus, where we perform our own basic synthesis and non-electron microscopy characterization. So I'm certain at this point in this workshop that you've seen many talks about transmission electron microscopy. I wanted to go over instrument geometry and configurations just a little bit, just by way of illustrating the imaging modalities that I'm going to be talking about in this talk, which are two. So as you are probably very familiar with at this point, in our transmission electron microscope, we have an electron source. Electrons typically go down and get a gravity boost. And then they encounter the condenser optics, which condition the electron beam before it hits the sample. The sample is actually immersed inside the objective lens, which is the main focusing optic or magnifying optic and focusing optic of the instrument. And then afterwards, we have the projection optics, and those are what determine which imaging or virtual imaging plane we are propagating to our detector. And the great thing about electron microscopes is that they are very tunable. Because our lenses are electromagnetic lenses, we can change up the configuration of our instrument simply by varying the current or even reversing the current sometimes in those lenses and changing the action of that lens. So some of our options could be for the condenser optics. We could have a parallel or focus probe illumination. And for the projection optics, we often switch between imaging in real space and propagating an image of a diffraction pattern, so reciprocal space image to our detector. And so today I'm going to be talking about parallel illumination, real space imaging, so your classic HRTEM, as well as maybe a more modern approach for diffraction that uses a focus probe. Okay, and so as I mentioned, today I want to talk about some of the supervised and unsupervised machine learning approaches that we are implementing and studying in my group. I'm going to touch on two different imaging modalities, as I mentioned. First, analysis of high-resolution TEM data. Then we're going to get into diffraction mode and talk about some unsupervised approaches that we are implementing. And then briefly at the end, I'll bring it together and kind of look forward to maybe fully automatic analysis that can be supervised using simulated data. Okay, so I'm going to dive in first to kind of a little vignette of using and optimizing machine learning protocols for analysis of high-resolution transmission electron microscopy, or HRTEM data. And what we're going to be looking at is actually the role of neural net architecture in image segmentation. So again, here is our imaging mode for this topic. We are in what I will call bright field conventional TEM mode. So this is an advantageous imaging mode for a variety of reasons. It is very dose efficient. You are collecting almost every electron that hits your sample. It is compatible with in situ or low temperature experiments where you might expect drift and things to be changing rapidly. It's a full frame imaging mode. One of the downsides is that these images contain a combination of amplitude and phase contrast. So the contrast in the image that's recorded on the detector is not directly interpretable. It's nonlinear, and we might not always know the degree of nonlinearity that is present. So this is a very typical image, just in case you're not familiar with looking at hundreds of these every day. This is gold nanoparticles on a carbon substrate. And one of our most basic tasks in image segmentation is going to be identifying which regions, which pixels in this image correspond to nanoparticles and which are only substrate. Okay, and so why do we want to do this? Which is ultimately, this is going to inform how we design our characterization pipelines, right? So local atomic scale information that you get from the TEM is critical to understanding nanomaterials. The TEM has really become a workhorse in synthesis science and for folks working in the field of catalysis, where you might want to answer questions such as, how does my catalytic activity change as I tune the exposed facet of my catalytic, in this case palladium, nanoparticles, as shown in the example on the far left? Or how might I tune the optical response of a nanomaterial just by changing its aspect ratio? So we really want to dig into these structure-function relationships, and to do that, we need to be sensitive to this local atomic scale information. However, there is a problem with nanomaterial synthesis and combining it with TEM, in that TEM, HRTEM analysis can be slow, especially if it is carried out manually. If anyone in here, I don't know if there's anyone here who works in synthesis, but you might know that you would have to go through many, many rounds of optimizing a synthetic protocol, and you simply aren't able to image all of your reaction byproduct. And in fact, you might be only looking at the success cases, and you might not have time on the instrument or bandwidth for your graduate students or postdocs to dig deep into what parts of the synthesis protocol yielded results that didn't work, that weren't optimal. And additionally, we also have the problem of heterogeneity in nanomaterials. Unlike in the field of biology, in inorganic materials, every nanoparticle will essentially be unique. And this can make it really hard to look at a broad population of nanomaterials and pinpoint what structural motifs are actually important for that optimized functionality. And so this is really the motivation for high throughput electron microscopy. And so we are lucky that it's an interesting time in, I would say, materials and physical sciences in general. Experiments are getting more and more automated and high throughput. And we are, even in electron microscopy, increasingly able to automate both our nanomaterial production, so sample production, and our imaging. And I will say this is kind of outside the work that we do in my group, but we really hope to benefit from this. There are some very, very smart people working on these problems of automatic synthesis using robots. Even at our molecular foundry, we have folks doing this, and automatically imaging and moving the stage and then collecting all the data. And so this is really the dream for a high throughput workflow. We would like to measure over a large parameter space, varying temperature, composition, and time, and maybe even do correlative functional measurements for all of these populations. Then we would like to image all of those populations and reduce our imaging data to some sort of useful statistical distribution, be it size, shape, aspect ratio, structure, composition, or defect content. All of these things are potentially useful information that we could measure in parallel, or even more interestingly, that we can observe how these distributions interact with each other, and whether there are correlations in our population. And that's when things get really interesting. Okay, and since we're talking about image segmentation, of course, we are going to talk about convolutional neural nets. Probably for this audience, I don't need to introduce or motivate this very much, but of course, CNNs, ConvNets have been widely adopted for analysis of natural images and are good candidates for analysis of scientific imaging data. And we're seeing kind of increasing adoption in the scientific community of convolutional neural nets. And really, I'm not going to dig in too much to the details of what a convnet is. I don't think I need to for this audience, but we're really taking advantage of the fact that CNNs are universal function approximators. And here our input data will be our images, our output predictions will be our classified pixels at the lowest level. So we want to determine what is a nanoparticle or what is our sample in our image and what is background. By learning, in this case, we're taking a supervised approach, so we will have annotated images for training data. Of course, I think that this is talked about frequently, but it's going to be a theme of this talk, so I'll go over it. Convolutional neural nets have been widely adopted for natural images. I think we all know this. There are distinct differences between scientific imaging data and natural images. So here are some sort of cute and broad examples of that. So natural images tend to have very large data sets that we all train when we identify stoplights in CAPTCHAs for Google to prove that we're not a robot. And are relatively cheap to label. There's kind of a natural orientational bias in images because humans tend to stand upright, not always, but a lot of times we do, and gravity exists on our planet and goes one way. The difference between natural images and scientific data is that often the scientific images are smaller, our data sets are smaller, and they're expensive to label because they might be more difficult to take, more difficult to interpret, and the training required for an individual to say that is a nanoparticle and that is background is much more high and uncommon than for a person to identify a stop sign. We also, especially in the case of TEM, we're looking top down. So we're looking at a flat sample, looking through that sample. It's a projectional image. And that means that they are rotationally invariant, which is not a feature of convolutional neural nets. And in general, we would like our output to be explainable for this application. And so going even kind of deeper and more specific to electron microscopy data, we need to think about the contrast mechanisms that are present in our images. So depending on the kind of microscope that we're using, its imaging optics, and the size of our sample, we can get a combination of different types of contrast in our image. And kind of the endpoints of this that I'm really alluding to are that in the low resolution case, we have majority of what we would call amplitude contrast. So you can think of this as when the beam strikes your sample, some of the electrons will scatter away and be removed from the imaging path. And that is statistically more likely to happen if I have a heavier material or a thicker part of my sample. So that's amplitude contrast. And analysis that might work well for amplitude contrast is thresholding. It's much more linear than the other kind of contrast that we might find, which is phase contrast. So in the high resolution case, when we have small particles, thin samples, and fewer aberrations in our imaging system, we might be in a scenario where phase contrast dominates and we are not modulating, not altering the amplitude of that electron beam very much, and the primary modification is in the phase of that electron beam. And in this scenario, we get nonlinear effects, and our images are much, much more difficult to interpret. And so our goal in this study was really twofold. We wanted to see in general how neural net architectures would influence our results, and we also wanted to detect if we might be unintentionally introducing any bias in our analysis with respect to different types of contrast in the scenario of both being present. Okay, so for this study, as I mentioned, we're mainly going to be looking at architectural constraints. In general, the most major architectural constraint that we have that we really can't change is that we need to use lightweight neural nets for this type of analysis. Our data sets will typically be 100 images. The biggest data set that a student in my group has ever labeled was 4,000 images, and I don't think she has forgiven me for that. So we are, and that's compared to 14 million annotated images for ImageNet. So we are always going to be constrained to a lightweight neural net so that we do not overtrain. And we're going to be using for this study a UNet architecture consisting of residual blocks, pooling layers, upsampling layers with skip connections. So The first half, of course, performs downsampling via a max pooling operation, and the second half will perform our upsampling. And for this part of the talk, I'll just be evaluating performance using the DICE score. Okay, so receptive field is a concept in neural net segmentation. It basically refers to the area of the input image that determines or contributes to the output decision of a single pixel. So receptive field has been shown to influence neural net accuracy in natural images, and it is also related to network complexity. Lighter weight networks actually have smaller receptive fields, because I think if you're great at reading equations with the fly, it's described down there, but conceptually you can think the more I downsample, the more I am including information from farther out in that original input image. So what my postdoc, Catherine Situ, did is modified a very basic classic UNet so that we could independently tune the receptive field and the network complexity and look at these individually. So she did that in a really simple way by changing our max pooling kernel. She also tested using a dilated kernel, and it works exactly the same way as the results that I'm going to show you today. So by changing our max pool kernel size, our downsampling step, we can tune the receptive field. We can make it bigger without changing the number of tunable parameters in the neural network, because that max pool size is fixed. So let's dig into some results. So by varying the receptive field, we can look at different contrast types and see what we get. So if we apply this approach to low resolution transmission electron microscopy data, we see no trend. So that's perhaps best summarized in the trend of hard DICE scores over there on the left. And you can also see pretty easily qualitatively, looking at these representative images, comparing them to the ground truth, that even as we make the receptive field quite large, we do not see any alteration in the performance. There's a small trend with the number of tunable parameters, which is to be expected. Okay. However, if we switch to high-resolution images of smaller nanoparticles, all of a sudden we see a strong trend. So here, as we tune the receptive field and make it larger, our performance increases. We still see a trend, a general trend of performance increase with number of tunable parameters, but within neural networks that have the same number of tunable parameters, as we change the receptive field independently, our performance gets better. So this is a distinct difference. Receptive field influences performance in high-resolution data, but not in low-resolution data. And this is just them side by side. So, oops, sorry, my citation is jumbled down there. So we attribute this to actually these differences in contrast. Receptive field seems to matter the most in phase contrast dominated images. And intuitively, I know it's a little dangerous to interpret how the neural net is working, but it makes sense that when the network needs to learn nanoparticle texture and cannot rely on thresholding or contrast differences between sample and background, that a larger receptive field would be necessary. So intuitively, this kind of checks out, and we can check this trend by looking at different size nanoparticles. We expect the degree of amplitude contrast to increase as we increase the size of the nanoparticles, and we see that indeed the trend is strongest when we in receptive field performance, that when we have small nanoparticles. So that checks out. So to summarize this, receptive field turns out to be something to think about when you are optimizing a neural net for analysis of HR to EM data. And increasing the receptive field without increasing the number of tunable parameters by using a larger max pooling operation or a dilated kernel, if you want to get fancy, can increase the network accuracy without increasing those number of parameters and risking overfitting. And again, this is getting into more intuition and just our own thoughts, but we do feel that sufficient receptive field, especially in that phase contrast dominated space, helps segment the difficult regions of nanoparticles where the texture, understanding texture would be important to segmenting those regions. Okay, so that was just a quick walkthrough of some results of our supervised learning approaches. And next I want to show some recent work that takes an unsupervised approach. And so we're going to switch imaging modalities to diffraction space. And I'm going to talk a little bit about how we have used an unsupervised clustering approach to interpret large nanodiffraction data sets, and how we have come to understand that the role of featurization choice matters, and show a consensus clustering approach that increases reliability and trustworthiness of this approach. So if you're not familiar, again, I think this has been pretty well introduced earlier in this workshop, but I'm going to be talking about 4D STEM, or really four-dimensional scanning transmission electron microscopy, which is a scanning nanodiffraction method that's illustrated conceptually here. So what we're doing is we're focusing our electron beam down to a very small region of our sample, and then we're rastering that beam in a grid, a 2x2 grid, over our sample. At every raster point, so at every grid point, we are collecting a diffraction pattern, so a 2x2 diffraction pattern that corresponds to every grid point in our scan. So that is how we get the 4D in 4D STEM. It's really a four-dimensional data cube. And we can really apply any analysis method that we want to the output diffraction data. Diffraction patterns have a rich variety of information from the same diffraction pattern. You can learn things like, what kind of crystal do I have? What is its symmetry? What is that crystal's orientation? Is it strained compared to the crystals around it? So really there are kind of a multimodal signal that you can get from a single data set. And the power of 4D STEM is that because we are taking our diffraction patterns from such a small volume, we're getting a very high resolution map of how these physical features vary in our sample. At least that is the goal. The hard part is that 4D STEM can produce very, very large data sets with thousands of diffraction patterns, and this makes them really difficult, and I would say in most scenarios impossible to manually analyze. With the advent of really fast electron detectors that have come on the market in the past 5 years, I would say, we're now taking terabytes of data in a typical experiment. So we can't do this by hand. Another problem is that we don't always know beforehand what structures will appear in our data set, or even how crystalline our data set will be. So one might think, and this is a valid analysis method that can be really successful in some scenarios, that you could template match or compare to simulated diffraction patterns of a known material. This works great in a lot of scenarios, but not every scenario. And particularly in my group where we study complex synthesized materials, it's not always the best approach. So we would really like an approach that doesn't assume anything about our sample structure. Okay, and so here's where we come to our unsupervised clustering approach that we've implemented for 4D STEM analysis. So the goal of this analysis pipeline is to cluster together similar diffraction patterns. We expect that intuitively, as material scientists, we expect similar patterns will appear as spatially localized similar regions in the material that might share a crystal orientation, a crystal structure, et cetera. So initially, we kind of embarked on this project with a goal of developing and testing an unsupervised workflow to cluster these similar regions together in the material. And so this is kind of a schematic of how the output of this might look, and how you might understand this data set. So I have some imagined sample here with 3 different regions of different crystal orientations. And if I look at every, again, location in this data set that I've taken, there's a corresponding 2x2 diffraction pattern, nanodiffraction pattern that I've taken. And they will be different across the sample, but they should be self-similar in the differently colored regions. This is the core of what we would like to automatically detect. So to test this, we generated a data set with these features. So we actually made 3 data sets of silver nanoparticles with varying size, approximately 30 angstrom, 50 angstrom, and 80 angstrom diameters of the nanoparticles. And as the size increases, a feature of this data set is that the overlapping crystal grains, so again, this is a projectional method, so I can have nanoparticles kind of sitting on top of each other or overlapping. This will increase as the nanoparticles get bigger. So we expect that there will be regions in our data set that have two crystal orientations represented. And essentially, we simulated a full 40 stem data set for each of these nanomaterial sizes or nanoparticle sizes using kind of available tools that were developed by Colin Ophis and others at NSEM. Okay, so here is our starting point for our unsupervised workflow. So in real life, we have some data collection step. For what I'm going to show you today, that is a simulation step. Of course, we would have to worry about calibration and pre-processing. If we have real data, we want to remove spurious pixels. We need to kind of compensate for maybe some instrument imperfections. And then we need to construct our feature vector. So our approach here is going to be non-negative matrix factorization. And so really our input into our NMF algorithm are going to be feature vectors that we have constructed ourselves from our data. So we need to reduce our data in some way, and we can either study individual featureizations or we can combine them together. We could also do a spatial and size refinement step if we wanted to. We have not done that yet. But the takeaway here is that we expect the feature representation choices to play an important role in our results. Okay, and so here, let's see, go ahead and pop that up. Okay, this is a complicated slide, so I'm gonna talk through it. And I apologize. Okay, you can see the very top there. Good. All right. So we tested 3 featureization approaches, and if these seem like out of the blue choices to you, it is because maybe you're not a microscopist. These are really inspired by existing approaches for physically meaningful analysis of our diffraction data, also with the mindset of we need to reduce down these 2D diffraction patterns into some sort of feature vector. So, of course, one of the really common analysis methods for diffraction data is to simply isolate these Bragg disks and basically register them in XY space. So we tried this. We call this Bragg disk features. And essentially, you can think of this as majorly downsampling our diffraction pattern and turning what is really a disk with an intensity and maybe some intensity variation inside to a single pixel with an intensity corresponding to it. We can also take this Cartesian to polar elliptical transformation. So we can go from a typical 2D diffraction pattern with Cartesian coordinates and transform that to this polar elliptical representation where the radial direction is on the y-axis and that angular direction is on the x-axis. So if you're not familiar with this, you can think of all the Bragg peaks that are the same distance from the center and therefore correspond to the same d spacing in the crystal are now kind of on the same horizontal line in this type of representation. And we tried 2 featureization protocols that employed this type of representation where we were binning over a range in the radial direction and over a certain bin width in the angular direction. So we're going to call that angular averaging. And we also just took the radial variance in that radial direction. So these are the 3 featureization protocols that we tested independently of each other for our clustering workflow. So here are the results. You can see the original class labels from the simulated data set on the left. AG1, AG2, AG3, that's just the different sizes of the silver nanoparticles from smallest to largest. And then we have the radial variance, the angular average, the Bragg disk featurization approaches, the results shown in the columns. And so you can really quickly pick up on the fact the radial variance approach does not work very well. It is significantly outperformed for all three data sets by, well, I won't say significantly for the small one, but it is outperformed by the angular average and Bragg disk approaches as shown in this precision metric. So this precision metric is the precision of just nanoparticle versus not nanoparticle. So it's a pretty coarse precision metric. And if we want to get a little bit more detailed, we can actually look at the distribution of performance basically for clustering the individual nanoparticles here. So looking kind of particle by particle or cluster by cluster, here we're using this true positive rate metric, which is really just the probability that a positive measurement will indeed test positive for individual nanoparticles. And we see, first and foremost, a distribution of performance from particle to particle. And we see a bit of degradation in performance according to this metric for the Bragg disk distribution. And this is because, in contrast to when we were looking at the precision metric and we were just looking particle versus not particle, here we're looking for correspondence. And what we see is that the Bragg disk protocol actually overfits, and where we have overlapping regions in our nanoparticles, that is represented as a distinct class using the Bragg disk feature representation, which is not the case. So we see performance degradation there due to this overfitting. Okay, and so here again is some more results from this same data set. So what you might have noticed in the previous slide is that we had a distribution of performance from cluster to cluster. We also see a very big performance variance depending on how we actually kind of implement our NMF protocol. So if you have used NMF, you might be asking yourself the million-dollar question, which is, how did she choose how many classes to use? And the answer is, we really didn't. We chose a large number, and then based on the self-similarity of those classes, we have a merge protocol. So we just kind of pick big enough and then have a threshold for merging. So what we find, though, is depending on some pretty small changes in that merging threshold and the number of classes that we select, actually, as well as just the random instantiation of our NMF algorithm, our clustering pipeline can successfully reconstruct the different crystal regions of our original data set pretty well, but there is a lot of variance in performance. And especially when we started digging into the details of, data set 1 versus data set 2, I think that what we realized was that it was always going to be very time-consuming, annoying, and basically not user-friendly to optimize these tunable parameters every single time. So what we chose to do is instead adopt a consensus clustering approach. So our approach here is to train many models and then actually find where the models agree and then take that as our final results. Okay, so here is this illustrated schematically. So we start with our many independent models. For this kind of systematic study that we're going through here, we used 4 different parameter sets, basically varying the number of components and the threshold to merge the components, as well as 20 different initialization conditions each. And we grouped the similar models by basically looking at the intersection of clusters. So first we start with we kind of identify a foundational group of clusters. So from one of these parameter sets and initialization conditions, there will be a model that comes up with the highest number of components, and we pick that. And then we go through, and for each cluster under consideration, we compare its intersection with the foundational set. And this is basically how we come up with our bins of what we will call similar models. And to get our final cluster for each cluster under consideration, we just take the average of all the clusters that live in the same bin that have some sort of intersection. And if we don't find one, we leave it as its own cluster. So that's how our consensus clustering works. And here are the results of that. So consensus models across the board perform better than the set average. In most cases, they actually outperform any model from taking an independent model approach. But in all cases, they outperform the average. And the two kinds of failure modes that we are really able to overcome by taking this consensus approach is parameter selections. So if you look kind of within one of these blocks of performance, then you can see there's a distribution described by the block and the error bars. You can see some outliers in some cases, but you always see that this, I'm going to show you, that these colored dots that are shown, these are the consensus model performance. So they always outperform, whatever your parameter selection initially was. We also see these kind of outlier points that we really attribute to just bad initialization conditions, where no knowledge of the sample was retained. So this consensus approach also makes our results robust to this just kind of random bad initialization condition scenario. What it doesn't overcome is the overfitting problem that we observed in the Bragg disk feature choice. So for example, in this data set where there's lots of overlap, we are not able to resolve that using consensus clustering. So featureization choice still matters. Your input to your feature vectors still matters, but a lot of these errors or poor performance that are just related to the variability and the difficulty of choosing that correct parameter set can be overcome using this approach. Okay. So to summarize, unsupervised clustering is a viable strategy to interpreting 4D STEM data and giving physically interpretable results. Here is some real data. So this is our high angular dark field image of a nanowire that adopts this really complicated twisted morphology. It is highly polycrystalline and disordered. And through using this unsupervised clustering NMF protocol, we are able to map out many of the crystalline regions in this nanoparticle. And then we are really primed to input this into further data analysis workflows. In this particular example, we're interested in finding some characteristic grain boundaries or characteristic twinning that we think is related to this twisted morphology. So it's not just that you can map out these distinct regions in your data set, you're also then very well set up to tack on different analysis methods to your pipeline and continue. The other thing that I will say is that we are not the only folks who are using unsupervised approaches. This showing right here is some really nice work out of Paul Alivasatos' group, formerly of UC Berkeley, now at UC Chicago, where they are applying unsupervised classification to nanomaterial shapes from TEM data. So this is a real space approach. So it's not just for diffraction data, provided you have enough data and a way, again, of featureizing your input data that works. It's also a really viable approach for HRTEM and other electron microscopy imaging modalities. Okay, so for the last bit of my talk, I think I have about 10 minutes left, but I'll try to leave some time for questions. I want to quickly go over a fully automated supervised machine learning approach that we are developing in our group. So this entails coming up with simulation pipelines for high resolution TEM data and also trying to understand the transfer learning and generalization kind of dynamics that arise when we try to interpret real data with models that have only seen simulated data. Okay, so simulation of electron microscopy data is really, I would say, a cornerstone of modern electron microscopy. You know, there is a long legacy in our field, definitely before my time, of folks developing physically realistic image simulation that takes into account the physics of the interaction between the electron and the sample in a realistic way. So that existed before I touched an electron microscope. What has changed, I would say, during my career is these simulations have become much faster. We have better computational resources. We're able to implement these now, maybe on your desktop or kind of a beefy workstation in minutes rather than days. One of the bottlenecks, however, is actually making those input objects. So if we want to simulate something, so I've got these schematics of kind of idealized gold nanoparticles plunked on top of a amorphous carbon substrate. We need to be able to actually generate these objects in a way that is physically realistic enough to work, so that our images actually are realistic enough so that we can train on them and then interpret real data. But we, at this time, still do not have the computational resources to do, you know, an actual MD simulation of 10,000 gold nanoparticles sitting on a substrate, which, by the way, the substrates in our samples also have millions of atoms, and they need to be different from image to image, or your neural net would just memorize them. So we took an approach of, we would like to make input objects that are realistic enough. We're not going to be able to relax the atomic positions. We're not going to try to do that. And to build a pipeline that then takes advantage of the simulation tools as well as other structure building tools that already exists, so that one could then say, I'm going to be doing a high throughput study of XYZ material. I expect, you know, a certain distribution of features and characteristics in my data set, and I will be able to generate 10,000 images and train a model using this pipeline. Okay, so large structure data set curation. Here are a little bit of details. So we ended up calling it construction zone, and you can find it on our GitHub. But basically, we take sort of a CAD approach where we would first like to generate regions of atoms that have the crystal symmetry or basically just the distribution that we expect for all the materials in our system. Then we would like to define our volumes and their relationship to each other while resolving any problems with overlap. And then we would like to generate this big collection of objects, handle any conflict resolution while keeping very careful track of the metadata in our objects, both from a standpoint of what is the size of my nanoparticle, for example. Does it have any defects? If so, what kind and how many? All of the physical structural information, as well as all of the image generation metadata, so that you have this vast library that's very well annotated. This is an example. So, and really the purpose of this example is just to show that my student, Luis Rangel da Costa, was able to write this Python package in a way that's very user-friendly, because what we're doing here is we're going to define our substrate thickness, the size of our nanoparticle. We can define an original crystal orientation and then add some defects, some grain boundaries. And this is this much Python code. So it's very user-friendly and really meant to be used in a way that allows you to loop through many structures and curate a very large data set. Okay, I'm not going to go over that in detail, but really our original goal here was to generate training data for supervised segmentation tasks. So we also, alongside every image that we make, store a mask that designates the boundary of the particle in the image to make training really easy. And then I have 2 more slides on this that are just going to be very kind of high level and showing some results and kind of where we want to take this. So first and foremost, we were interested in knowing, did we create a protocol that allows us to train models purely on simulated data? So this is a model, again, trained on purely simulated data sets. We have some different types of structural augmentations. We're in the orange models shown in the performance histogram, actually including much smaller nanoparticles, and then varying the substrate thickness because that is a physical reality of our samples. So we are actually able to train several hundred neural network models and look at performance characteristics using this approach and directly apply this to experimental data. And we see that we can get very high performing very high performing models just from a simulated approach. By the way, this is all on a lightweight unit, very similar to the results that I showed in the first part of this talk. So this is, sorry, okay, 5 minutes. So we're kind of in the scenario that we were at with a consensus, or before we implemented consensus clustering, where you can be certain that one of your models will probably be pretty high performing on your data set. And now the real work of this project is to guarantee that that happens. One of the very interesting things to look at is the actual generalization dynamics. So if what I'm showing here are performance as you train over about 25 epochs. And what we're doing is we're training only on simulated data still, but we are just tracking how that model would perform on a validation set that is experimental. So there's no training input from the experimental data set here. We're just watching kind of sneakily how this performance changes. And what you see is the performance saturates on the simulated data set really quickly, but the performance on the experimental data set, which is the non-dotted lines, lags behind. And so now that we have the ability to test this with this high throughput image simulation and generation pipeline, we're poised to be able to really understand these generalization dynamics, I would say. And we really anticipate that this will help us optimize our training protocols for more robustness. And then, of course, and so we can really dig in to things like augmentation. I'm not going to go through all of these different performance histograms, but we're just tracking the F1 score for a variety of structural augmentations or image augmentations. So things like rotations and really how much the experimental data that we see. And we see some takeaways that I think are similar to what anyone who's studying transfer learning would see, is that you get a performance boosts the more you transfer learn, but you then in a way are overfitting to that data set that you're learning from, that experimental data set. So you're sort of zooming in and trading, I would say, generalizability for performance within that specific domain. So depending on your application, maybe those are some choices that you have to make. Okay, so that was a little maybe forward-looking chat about our upcoming work on simulation pipelines that we hope will alleviate a lot of the struggles that we have with small data set sizes. But I think that you can get a sense of how much work there is to be done before these can really be turned into robust, reliable tools that non-domain experts can use. But we're excited about it. I think it's a really fun space to be working in. So with that, I will conclude. In general, it's an exciting time to be an electron microscopist. Electron microscopy is becoming more and more high throughput and automated, which means that we need these advanced methods for information extraction from these big data sets that we cannot manually analyze. However, once we are able to do this, electron microscopy, I think, will be able to be incorporated into these high throughput materials design projects in a way that has not been possible before. So that's really exciting. And I'll just acknowledge my wonderful group and our funding. There's our GitHub. Check it out or email me. I will also mention that when I wear my staff scientist hat, the Molecular Foundry is a user facility. So it is free to use if you have an accepted proposal. So if any of these topics are of interest to you, feel free to reach out. It's very typical for prospective users to email and chat with staff scientists. Our next proposal deadline is spring 2023. So that was just a quick mention, and I'll leave up my acknowledgement slide, and I'm happy to take any questions if there's time.